Novak, welcome to Bendiksen Airways.
Yeah, thanks for having me.
I came across your name and your organization, Revive and Restore, just recently, and I also read the Time Magazine article on colossal science direwolf. But before we jump into this, how would you introduce yourself and what you do?
My job title is the lead scientist of Revive and Restore. Revive and Restore is the world's first and premier nonprofit organization, so totally philanthropic, dedicated to advancing biotechnologies for conservation in nature. Um, our tagline mission is genetic rescue for endangered and extinct species because we believe that biotechnology covers a large umbrella of applications in nature. We currently fund over 50 active projects around the world on every continent with institutions working with a number of both endangered, common species towards the goal of helping species in their environments. And we do also work with a couple extinct species to try and recreate those. That's what we started with, and I lead the passenger pigeon project, and I've published extensively in both peer review and in press like this about the developing practice of de-extinction and what it really is and what we're trying to accomplish.
Nice. There are a handful of these organizations working on this now. Is that correct?
Revive and Restore and the company Colossal are really the two main entities. Rewild Europe and some other groups, I believe, have the Toros project. It does not include gene editing at this time. It's more of a breeding project. There is the Quagga project in South Africa, which is another breeding project. And Michael Archer and some others in Australia are working on the Lazarus project to recover an extinct type of amphibian down there. They're working with actual tissue from that animal that was frozen, so they're bringing back the actual animal. So you can view it as de-extinction, or you can see it as saving a species through literal resurrection. So there's a few entities, but not very many. The majority of the projects are colossals and revive and restores.
Okay. So, what inspired the founding of Revive and Restore?
So we were founded by a pioneering environmentalist and techno optimist futurist, Stuart Brand, and his wife, a serial entrepreneur in the medical world, Ryan Phelan, who is our executive director to this day, and the powerhouse behind our organization. And what inspired it was in 2012 the emerging technology of CRISPR-Cas9 made it clear that the idea of changing animals' genomes in a way that we'd never been able to before at cost and scale would make it possible to maybe start reviving some of those extinct traits from extinct species. And Stuart Brand had a high interest in the passenger pigeon and decided that he would create a nonprofit around that concept of recreating the passenger pigeon. I was hired on right away because I'm a passenger pigeon expert. In those early days, I was working on their genetics. As a graduate student, I left grad school and started working for them. And, you know, I think we all had hoped early on that the mission of de-extinction would be something to kind of push the field further, which it still is, but we had kind of thought that conservation was likely adopting some of these tools of genomics and in vitro sciences more like medicine was and agriculture was. But the truth was is back then, and even really to this day, most people in conservation are still not really using genomic science or technologies. And so we very quickly, rather than just doing passenger pigeons, started to work heavily in promoting these technologies and developing them for other species and living species. In 2013, we started working with the black-footed ferret, which is on my shirt here, and slowly built up our mission over the years, and and now work almost exclusively with living species in their ecosystems.
Right. Why bring back extinct animals?
Well, technically, people have been doing this for over 100 years. So when we think of it in terms of like, oh, woolly mammoths are extinct and they've been extinct a long time. But species also go extinct in local areas. So, a good example are beaver in Great Britain that were killed off on the island 400 years ago, but there are still European beaver throughout Europe. And people, as they recovered them in Europe, started thinking maybe they could reintroduce them to Britain. And they have. They've done experimental sites throughout Britain, from Scotland down through England, and I believe even Wales. And that's a species that hasn't lived there in 400 years, and now it's back. In Spain, a team of conservationists have reintroduced European bison, which have not lived in Spain for 15,000 years. And these species still survive in other places, so they're able to take them from where they've survived and put them back where they once lived. They don't have to recreate them using biotechnology. But when you think of in terms of reintroducing a species to a habitat, people have been doing it all around the world for over 100 years now to restore the missing roles and damage done caused by extinctions. And biotechnology really just opens up the ability to take what people have already been doing for a long time and go further to species that have no living populations and no living relatives to use for those purposes. And so it's to me it's really the same practice. We're just introducing these new technologies, and we cannot recreate the original extinct species, but we can revive some of their genes in a living relative to get those key traits so that a living animal can resume the ecological role and the evolutionary function of an extinct species. And that has not happened yet. We are still some years away from getting to that. But I do believe it will be successful, and it's coming pretty quickly. I mean, that is part of the big news from the colossal dire wolves, is that this is work that's coming quickly for some species. For the passenger pigeon work we do, birds are much harder to advance this technology for. Species like amphibians and reptiles, we have really no technologies to do this for. For most invertebrates, we don't have the technologies to do this. So it's going to be a continually growing field, definitely worth investing in, but the early wins are going to be just a few very, very particular species.
Must be exciting work.
I certainly think
Um,
so. Yeah, I've wanted to do
yeah,
it since I was a kid.
yeah, exactly. But but the dire wolf, how how close is it to the, let's say, original? I've seen some criticism of just making some gene editing doesn't make it a dire wolf.
Well, that is correct. So, and as the International Union for the Conservation of Nature, of which Revive and Restore is a member, published in 2016, de-extinction is not recreating the original animal. It is creating something that's functionally equivalent. And that's why projects like the Quagga Project or the Aurochs Project qualify, because they're not reviving any of the actual genes of the extinct species, but they're breeding something that fits the form and function. I think the projects that introduce gene editing give a little bit more of that biology into it than breeding can accomplish. But at the same time, you know, maybe editing just 14 genes is not enough to recreate the direwolf. Maybe editing only 3 or 4 genes is enough. At this point, no one in the scientific community has seen the data, and these wolves are also too young to really make those assessments too. They need to finish growing up. We need to measure their bones, measure their traits, do a lot of science here to really evaluate if they qualify as what could be a de-extinct dire wolf. However, the IUCN, which put forth that agreed upon definition, did issue a statement just yesterday that from the IUCN definition of de-extinction, colossal dire wolves are not dire wolves. They are just an example of several gene edits in a gray wolf, and there is no evidence as of yet that any of those edits are creating traits that are actually different than any of the traits we see in the many types of gray wolves we have today.
Okay.
And that was a
You
statement
s-
made by the IUCN Canid Specialist Group, a panel of people whose entire careers is studying wild canids.
yeah. You said earlier that some are working with frozen tissue to bring back the actual animal. What
Yes,
is? Yeah.
that is
Go ahead.
the Lazarus project that is headed by Michael Archer and others, and that is gastric brooding frog, um, which is a really unique and incredible species of frog. It the males swallowed the eggs and turned off their stomach acid and incubated and raised their babies inside their stomach. Um, so a really unique trait, and a scientist that studied them nearly 50 years ago had a couple carcasses that he put in a freezer, and people found those decades later, and they were able to get actual live cells from those carcasses. They've made some embryos, early embryos, um, but there are some barriers to getting those cells and those embryos to grow the whole way. So they know that they can get the cells to live and divide, but they've got some more work to do, and breakthroughs in their work will certainly help other living amphibians as well.
Yeah. What other kind of frozen tissue or old tissue is around that can be used potentially?
Well, really, the only tissue that's really good are tissues that are not just put in someone's freezer in their house, but in what we call cryopreservation tanks in liquid nitrogen. Um, People have been keeping liquid nitrogen tissues since about the 1970s for wildlife in various places. We don't have a lot, but people have been collecting more and more. There are great examples like the seed bank at Svalbard, where seeds can be kept at low temperatures, dry or cryopreserved. So, there are some really great examples, and we at Revive and Restore we work with a species called the endangered black-footed ferret. They are endangered. They're still alive. They got down to just 7 unique individuals and have been bred back into existence over the years. Um, and that was in the 1980s. And someone had the foresight to keep some tissue from one of the female ferrets named Willa, and she was not one of the 7 that they're of ferrets that they were descended from. She was an 8th individual, and because they made a cell line and froze those away at the San Diego Frozen Zoo, we were able to, with our partners at Fish and Wildlife Service, who work to save that species, and Viagen Pets and Equine, commercial cloning company here in the U.S., and San Diego Zoo with those cells, we were able to take those cells out of the freezer, send them to Viagen, produce embryos, and literally revive that animal. We have 3 clones of that animal, and last year one of those clones successfully bred and gave birth, and so now we have 2 baby ferrets that have new genetic diversity in the population, and that's completely because somebody saved those tissues decades ago.
Yeah, interesting. How does it affect the ecosystem if you bring species either extinct or not to the environment they haven't been in for years? How does that affect the ecosystem?
Yeah, that's a question we've had at Revive and Restore for some time, which led to some research I did around 2019 to 2020, and we published a paper in 2021 that's specific to the United States, partly because a global study was just really too big. But as I said, people have been reintroducing local extinct populations for a very long time. People have been moving animals around to restore populations for a long time. So we wanted to see, given that this has been going on for decades, has it ever gone wrong? Has it negatively affected the environment? And in the US, over the last 125 years, we couldn't find any significant examples in which restoring wildlife populations, even sometimes centuries after they had been gone, had led to ecological harm. And one of the reasons that makes that different than, say, when we hear about invasive species that are non-native and cause harm is that people have been primarily working with, reintroductions and translocations of native animals. And that's because even if a species has been extinct for many decades or centuries or even thousands of years, all of the species alive in an environment today, the native species at least, once lived with that extinct species for hundreds of thousands, possibly over 1 million years. And they co evolved with them. And they developed relationships and traits that work together. And so you can plug that extinct species right back into the ecosystem because those traits match up. When you bring in an alien species that doesn't fit in that ecosystem, it just doesn't work all the time. Sometimes non-native species do work because they are more of a fit, but invasive species cause big problems. And what we found is the history of species that are invasive that cause harm were perpetrated by private individuals, companies, government agencies that had nothing to do with wildlife and conservation, but were almost exclusively had to do with agriculture or other human economic incentives where they didn't consider how what they were doing would affect the environment. Whereas conservation organizations that have been working for over 100 years, including government conservation organizations and nonprofit companies and even for-profit partners in conservation, have worked according to risk assessments and guidelines, working in the ecosystem. And it's just a very different way of thinking and practice that makes the difference between a history that's been very positive versus one that has created ecological disasters. And in de-extinction, our plan with Revive and Restore from the start has always been to be a conservation entity, to work under those frameworks and what we've learned from conservation so that hopefully someday when we go to try and put a new type of passenger pigeon back in the environment, it plugs right back in like the original would and works like a reintroduction and not like an invasion.
Exactly. When you have when there's a new species born or bred, how long from its birth to when you potentially reintroduce it to the environment or to the wild?
Well, it can be as quick as a few years, and it could be decades. A really great example is the black-footed ferret. And other species that were saved from extinction from breeding. So, the black-footed ferret and the California condor here in the U.S. Were taken out of nature both in the year 1987. The last individuals were brought into zoos and breeding facilities, and so they were gone from the environment. And they only had a few of those animals left. So they started with a very small number of animals. But within about 5 years, they were able to breed enough offspring to start reintroducing back into nature. So, I mean, that was pretty quick. Other species have spent longer. So recently, the Guam kingfisher is great. This is a great conservation story. It lived on the island of Guam. The arrival of the brown tree snake, one of those alien invasive species, obliterated the wildlife on Guam. People captured some Guam kingfishers, brought them into zoos to save them, and they spent over 40 years in zoos, unable to ever go back to Guam. And that's not because they couldn't breed enough of them, but they had struggles there. But they had to figure out where could they safely put them back in the environment that wouldn't cause harm to other species. And a neighboring island called Palmyra Atoll was recently restored. They got rid of rats and other invasive species. And while the Guam kingfisher never lived there before, it has the same types of trees, plants, and other vegetation and environment that the Guam kingfisher can use. And it was deemed that it would be safe to put the Guam kingfisher there. Several years ago, Guam kingfishers finally went back to the wild after over 40 years being in captivity. And just this year, within just the last few weeks, it was seen that they have started laying eggs in the wild. So, it all depends on the environment, how well the animals breed, the type of care we can provide to them. But the history of conservation does show that you can start with a small number of animals and rapidly get animals back into the environment. Sometimes it takes longer. And even if you get them back into the wild, it does still take more decades to get them back up to the numbers where they can really sustain themselves and start working in the environment in a healthy way. But there are great examples of success with that as well. Here in the United States, the bald eagle is a great example where in the 1960s, there were fewer than 1,000 bald eagles left. And that was due to pollution like DDT and habitat disruption. And they never had to really bring those into zoos, but just by protecting them and cleaning up the environment led to them fully recovering. There are over 150,000 bald eagles in North America today.
Nice. When will we see? When will we see a dinosaur?
Oh, hopefully never. so, so with what we're doing for passenger pigeons and what other entities are doing, like colossal for mammoths and dodos, we rely on sequencing the genome of that actual extinct species. And you can get genomes from the permafrost of northern Europe and Asia and Canada, and that from samples that are 100,000 or 200,000 years old. The oldest DNA we've gotten that's really any good is a full genome of a horse from 700,000 years ago. But people have been able to get small segments of genomes from a mammoth at 1.2 million years old. But that's because there are frozen layers of permafrost that have been frozen for 1 million years. And it's that freezer effect that keeps that DNA from fully degrading. The science has really concluded that, under most natural circumstances, DNA degrades to such terrible small bits and mutated forms after about 1.5 million years that it's just not really recoverable. Well, dinosaurs went extinct 65 million years ago, leaving only their dinosaur bird cousins today. And so, using the bird today, some scientists have recreated dinosaur traits in embryos. They've created a dinosaur snout, dinosaur ankles, dinosaur like claws, longer tails, even dinosaur like feathers. And so, possibly, someone could take a living chicken and make it look like a tiny little raptor dinosaur from the Mesozoic. But it's just a look-alike.
Yeah.
It has no original information from the DNA of a dinosaur. And that's very different from what we can do with, say, a passenger pigeon, where we have sequenced the genomes of of now 18 passenger pigeons from the year 1860 to 1890, from museum skins, from skins and feathers. Like here, I've got a couple feathers of passenger pigeons right here. And you could take the tiny bottom of that feather and sequence that animal's genome from 150 years ago. And then we can actually understand its mutations and resynthesize those and put those in a living animal. It's not that simple. There are technologies we still need to advance, but we do have passenger pigeon genomes. There are mammoth genomes. There are genomes of other extinct species from about 100,000 years ago to present. That's really the only time frame where any of this is even really feasible.
I'm guessing this is a pretty high risk, high reward project. How do you go about evaluating when a project is worth pursuing or not?
Well, at Revive and Restore, we like to go along the lines that also the IUCN has published on, where we evaluate, the importance of bringing back a particular species for the environment today, not simply just because humans caused the extinction. So you look at an environment today and say, okay, are we missing a key predator? Are we missing a grazer? What are we missing in this environment that would help species today be more prolific and create more productive environments for restoration? And that's the first kind of step going towards. Then we say, like, okay, for the passenger pigeon, there's a clear value to the forests today, but can we get its DNA? So then we have to go to the specimens and go, okay, can we get genomes? Is there a living relative that we can actually gene edit and recover those traits that would work? Can we care for that animal in a zoo or a lab environment so that it has a good life and also we can be successful? So there are a number of considerations from the very start of an idea all the way to restoring in the wild. We have been doing a lot of ecological research on passenger pigeons, and we're still doing more today. We're going to start experimenting and resimulating their role in the environment to study the effects of the environment. And we get to do that for passenger pigeons because we have these books from the 1800s where people compiled records of where they lived, how they lived, and we have more information to kind of understand that restoration phase. And for most extinct species, and even most living species, we don't have the information of their ecology to really understand what we're doing. So I think, these early projects with species like passenger pigeons, dodo birds, and other species where we can have some of the better science, will not only advance the biotechnology, but really make a better practice for doing this more often so that someday we could bring back species where we know less about, but we believe have a valuable place in nature. And hopefully someday we've restored so much nature and gotten to a point where we exist with nature so well, and we've don't have any more big endangered species problems, where we really could start bringing back more and more of the species that humans have killed off in ways that reproduce them in the environment. So
Mm.
that's it's a nice idea for the future. Right now it's just we work on those species that could have really, really big beneficial impacts.
How far away is potentially a woolly mammoth, and how would it fit into today's environment, do you think?
Well, the company Colossal has publicly stated that they plan to give birth to a new de-extinct woolly mammoth, or a cold adapted elephant, as they say, in 2028. That means they have to be producing embryos in early 2026, and putting those into surrogate mothers. Now, given the advancements in the field, and the fact that they're working, the woolly mammoth is a mammal where the most technology is more advanced than in birds or other species, they could be on track for that timeline. It's But it's a 2-year pregnancy, so that's why they have to get their embryos into mothers in 2026 to meet 2028. And the pregnancy part of this is a challenging phase. Natural pregnancies can miscarry and be lost. When you start using embryos made in vitro, the success rate goes down, and so it takes a lot of surrogate mothers.
In vitro, does that mean in the lab?
Yeah, when you make embryos in the laboratory and then place them into mothers, the success rate of producing births goes down. And so you need a lot of surrogate mothers. When it comes to elephants, they can likely only put one embryo in a mother to be safe. So in horse cloning, we've at Revive and Restore, we've been a part of also cloning the endangered Przewalski's horse with San Diego Zoo, and those results were very good. But for the safety of the mother, they only put one embryo into the surrogate mother. That's because if they were to put more than one embryo and there were twins, twins pose a risk to the mother. This is a species that only has one baby at a time usually. Elephants are the same way. They usually only have one baby at a time, so twins and triplets would pose a risk. In our black-footed ferret cloning, we put between 25 and 30 embryos into the surrogate mother because they can have litters of anywhere from 2 to 4, even 8 babies. Now, only about one of those cloned babies ever ends up really becoming a baby, but we put in a lot of those embryos to guarantee that success. So with the elephant, it's hard to say how many embryos and moms will be needed. It could be possible to reach that by 2028. It could be possible to do with just a few dozen gene edits. It might not have to be thousands. I do believe for our passenger pigeon work, We are working to make advancements in cell culture and reproductive technologies and gene editing for the species that I believe will allow us to start doing our gene editing work in 2029 and possibly produce the first babies by maybe 2032. So the bird work is a little more challenging and a little different. Once our surrogate parents are laying eggs, it will only take 14 days for those eggs to hatch, but it will take a couple of years to get to that point. So sometime between 2032 and maybe by 2040 is when we'll hopefully have birds that we can show the world that look and behave like passenger pigeons and will eventually be able to fill that ecological role.
I've read a lot lately about the small bird population dramatically decreasing. Is that correct?
Here in North America and a lot of places in the world, there has been a severe outbreak of avian influenza for the past 2 years. So a lot of species that have not dealt with avian influenza, a lot are being impacted. There have been mass die-offs for a lot of species, particularly water birds. This is an example where biotech solutions could be big, but also they're just hard to implement at scale. Things like vaccines would be helpful, providing wild birds with the genes they need to be immune to these diseases, or coming up with ways to use biotechnology to try to get rid of this virus on the landscape are going to be very important. Luckily, for some endangered species, a vaccine has been developed, and some species are showing that they're not dying off due to the disease. But it is a very serious concern globally. And disease in general is a huge concern globally for endangered species. As the climate changes and more invasive species and more global trade just keeps connecting the world, pathogens are spreading rapidly. The COVID-19 pandemic is a great case of that. That disease spread across the world in less than a month. And now imagine hundreds of diseases affecting thousands of species other than humans. That's what's been going on for decades now, and it's just getting worse.
Could you walk us through your three-pronged approach at Revive and Restore?
Our three-pronged approach. I've never actually heard it called our three-pronged approach. Um,
That's what I read.
Yeah, yeah. I'm going to have to read our latest webpage, but there are three things we do that we talk about. So, um, what we do at Revive and Restore is we work to advance new technologies. So things that have never been done before that are we would not be able to do with a wild animal yet. So we work with lab mice, domestic ferrets, things in the laboratory, or cell cultures, things to make completely new tools. Then we also then try to implement biotechnologies. So once a technology is ready to start using on wildlife, we want to see that happen. And there are a number of technologies right now that are ready to use. The most basic is simply sequencing genomes and using the insight from DNA to make better decisions about how to protect species, where to protect species, how to restore their populations, even monitoring them using things like environmental DNA assays. There are so many tools through just genome science alone that can help conservation. Over 30 of our 50 projects we support right now in the world work just in the realm of genomic information because it's such a powerful tool that we need to use more of. We also have our cloning projects with the black-footed ferret and the Przewalski's horse. Cloning is a tool that for some species, particularly mammals, is a tool that could be used more. Especially with biobanking. So biobanking is something we've been working with Fish and Wildlife Service and other partners to get more tissues of endangered species saved away for future purposes. So that's where we're implementing the technology. But of course, advancing the science and implementing technologies, no matter how many teams we fund and work with, really don't amount to much if we're not also confronting the socio-economic and political nature of conservation. Conservation that happens in nature is done by people, landowners, public constituents, governments. It is an immensely complex social endeavor. And so we convene meetings of biotechnologists, conservationists, ethicists, lawyers. We bring people together at meetings to talk about these things, to discuss these things. We've published special issues examining these issues in the past, op-eds, other things like that. So communications and advocacy are a big part of what we do. And so that's the three-pronged approach. Advance the science, use the science, and convene and build the community to get the engagement out there and show the world that these are technologies we can believe in and we can pave the way to use.
Yeah. Because how difficult is it to work with the policymakers? I'm guessing there are a lot of laws and regulations in place.
It can be very challenging. Our first big win in that realm is with a species called the horseshoe crab, which live in China and the eastern United States. They've been on this world for 450 million years. They've survived 5 mass extinction events as a group. And yet today, because of humans, they are becoming endangered. And they're so important for humans because years and years and years ago, decades ago, it was discovered that their blood, this really amazing blue blood, has compounds in it that can detect deadly bacterial endotoxins in water and other substances. So every pharmaceutical company in the world, every medical company in the world for the last 60 years has used the blood of horseshoe crabs to ensure that the medical products they create that go inside the human body, vaccines, hip implants, insulin pumps, dialysis, and also the water they use in their laboratories developing their science are free of these deadly endotoxins because you know what? No one wants a medical device put in their body and then get an infection and go septic and die. So everyone alive today that has gotten a vaccine or any kind of medical procedure like a surgery literally owes their life to the blood of horseshoe crabs. Well, back in 1995, a brilliant scientist, I'm sorry, I've forgotten her name, but this brilliant scientist found the gene in the horseshoe crab that creates the compound that is used in that. She took that gene, synthesized the gene, put it into bacteria, and just the same way we make insulin today, was able to harvest that protein and create the synthetic alternative. And that was in 1995. That was 30 years ago. And the company Eli Lilly adopted it, but there were huge regulatory hurdles for them using it. So here in the United States, the FDA, the Food and Drug Administration, has to approve what can be used in medical science and pharmaceutical development. And a nonprofit third-party institution informs the FDA called the U.S. Pharmacopoeia, the U.S. Pharmacopoeia. And they felt that there wasn't enough science to support the use of the synthetic alternative for many, many years. Well, then it became clear that there was also pressure from the companies that go out and harvest and bleed horseshoe crabs to fight against the adoption of this. So, it took over 20 years to get this approved. But the incredible thing is that thanks to a coalition of organizations that was co-founded by Revive and Restore, a landmark review study by Revive and Restore, and advocacy from Revive and Restore, and these 40 other institutions that made this coalition, investigative journalism uncovering the pressures from the for-profit bleeding industry, and the help of companies like Eli Lilly, the U.S. Pharmacopoeia finally published their chapter paving the way for this synthetic alternative. And on May 1st, that will go into full effect through the FDA, that companies will be able to use that without further regulatory work. For new drugs, there will be some work for legacy drugs for them to adopt it, but it's going to potentially change and revolutionize the field and make it to where we don't have to keep harvesting a wild animal to support human life. So that's been our big win in that arena. And it was challenging. It is so many constituencies to deal with. And I think, a lot of people face that with other conservation interventions that aren't even biotechnology related. It's always a complex conversation when it comes to nature and anything in human society.
Exactly. When you work with such large timescales, how do you, number one, motivate yourself, and how do you measure success?
Well, yes, when I started this work, my hair was brown. um, And now I'm I was a fresh, I left graduate school to start the job, and now I'm a father with 6-year-old boys, and I've been working at this for 13 years. It took 7.5 years to build the relationships and science and iterative permits necessary just to clone the black-footed ferret. And we're still working on science for that. So yeah, we're 10 years in on a lot of our projects, and staying motivated is, in to be honest, is for me is getting the chance to talk with people like you, knowing that what we're doing will reach new audiences and inspire possibly young people and other people to get involved. Sharing the work is huge for me as a motivation, but also having those big wins when the first black-footed ferret clone was born and was healthy. It was just a huge moment. I personally drove my family out to see the ferret because I don't do the cloning work. I do the science from a computer. The people at Viagen made those embryos. The heroic veterinarians at the Ferret Center that have been caring for black-footed ferrets for decades brought those clones in the world. So I drove there to go see it, and I held Elizabeth Ann in my hands, and that kind of, made me feel more motivated to keep doing that work. We've had some more recent successes that keep me going, that hopefully in the coming months we get to talk more about. And working with incredible scientists keeps me going. But there are tough days. There are really tough days where things seem bleak, and there are days where I've thought maybe we're just not gonna have success. There are days where I've thought about quitting. And what's amazing to me is a lot of times just by chance on those really tough days, I'll get an email or a call from another scientist or a student or someone that tells me how much they believe in our work, and that's really all I need to keep going. Because
Yeah.
speaking of mammoths or passenger pigeons or dodos, we might plan on having the first new generations in the next decade, but it will take over a century to really restore those animals to the wild and achieve the goals we want in the world. It will take decades to change how we farm food and obtain energy. And we need to work faster because of climate change and other issues. But it will take years to accomplish these things. And for me, I don't do this because I want to see these things happen in my lifetime. I do. I really do want to see them happen. But I do them because these are the types of things that are going to make a better world for my kids and their grandchildren and people in the future. And so
Mm.
there are so many people that have not yet been born into this world that deserve a world that is healthier and has more life in it than the one that we've inherited. When I was born, there was no passenger pigeon. When my sons were born 6 years ago, in my lifetime, hundreds of species went extinct. Everyone born is born to a world with less diversity and less richness in it. And I think if we really work hard, we can stop that, and new generations can be born into a world that has more in it rather than less.
Do you see your work as a part of a larger movement to rethink humanity's relationship to nature?
I absolutely hope so. I believe it is, and I hope it is part of a bigger movement. For the passenger pigeon especially, it was once the most abundant bird on the planet. There were billions of them, and they we're in these huge flocks. And I've met many people today that fear the return of a billion birds overhead. They fear the idea of life being so abundant. And, for me, that's why this is such an incredible, inspiring project, because it will take a long time to have a giant billion flock of passenger pigeons. But if we as a society can have the comforts of our modern life with billions of passenger pigeons, millions of bison, and countless other populations of species thriving, that's achieving real coexistence and success. But the baby steps to get there are really re-examining how we just live in nature right now and care for the species that we still have. And I believe we have every technology and every ability to live with nature in our cities, in our communities, and to restore more wilderness and use less land for our needs. We just have to make it a priority as nations and as a world to make the investments to change.
How do you see the ethical issues?
Well, ethical issues, with many things, it comes down to how we do these things. I believe in biotechnology, we have to be as transparent as possible with our motivations as to why we do this work, the economics of the work we're doing, and the science behind what we're doing. We need to communicate clearly the science. I also believe we have an imperative to only talk about the science when we understand it and prevent false narratives from ruining the conversation. We have to do really important science and then communicate it well. And we have to work with a lot of people from many different walks of life to ensure that what we're doing not only accounts for the potential risks and unintended consequences, but thinks through what the intended consequences and benefits could be and the benefit sharing to the people involved, to ensure a legacy that is beneficial to humans and nature. And also, basically, just works with a moral compass that we can agree is something we care about in the world right now. There are a lot of ethics to consider, and it really comes down to how we work on best practices. So I think we can provide in this work, I think we can do it ethically and we can do it responsibly. We can provide welfare for the animals, plants, and the human beings involved. It takes the extra commitment and the extra work to make sure to do things well without taking shortcuts and working with government oversight and governments that can be trusted in the work. But building trust and doing things according to an ethical framework, absolutely, we can do that.
Nice question on this. If you could bring back, let's say, two or three species that you would like to see, no matter the difficult technology, which would it be?
Oh, well, one, of course, is the one I'm working on, the passenger pigeon, which I think is a really great candidate. I believe, I'm in full support of the dodo projects, that project that's happening, and the thylacine project. But more personally, I would like to see the restoration of some of the large moas of New Zealand, which could be really helpful to their native plant life. New Zealand has lost a lot of species, so as Hawaii, their honey creepers, species that pollinate certain flowers would be great to get back. And once again, these new forms that we gene edit, right? We were not really getting back the originals, so but we can reproduce that organism in a legacy form. And yeah, I think those, I tend to be a bird lover, so birds like the Carolina parakeet, the great auk, which lived in Norway and northern Europe, would be great to have back and would have a place in today's ecosystems, that could benefit and thrive. So yeah, it's hard to think of species from the last couple thousand years that I would say do not deserve a chance to be restored. But the reality is there's some species that we don't know very much about. one of the species I would love to see again would be the Choiseul crested pigeon. It's one of the most beautiful pigeons in the world. Lived on Choiseul Island in the Solomon Islands, but we know very little about it. We don't really know what it ate or really where it lived. We don't know a lot. And so we would have to do a ton of science to try and figure that out. Maybe someday we will be able to responsibly do that and do it effectively. But it's really when we start thinking about species that died out possibly tens of thousands of years ago where things get more complex. Whereas we don't know as much about their environments and their ecological roles to make the right decisions. And there's other things to factor in too. When you think of restoring animals, you can't restore a predator unless you also have their prey. You can't restore a pollinator if you don't have the flowers that it once pollinated. So the mutual relationships have to be there. And so there's a logical sequence. And I think when we think of species from tens of thousands of years ago, the best ones are things like the giant herbivores, animals that can eat all the plants we have today and create that huge impact in nature that their large bodies and major migrations had. It'd be exceptionally challenging to reproduce something like a giant ground sloth, but we know that there are plants alive today that benefited from the diet and interactions of those giant sloths. So perhaps if we can create synthetic uteruses and artificial systems to give birth to those babies, we could create those animals and they would have a place today. Woolly rhinos would be of benefit, these large bodied animals. And then, you know, then there's a lot of other species that could play a good role. But but yeah, it's an incredible future we could have, but it's going to build more slowly. I mean, the science is coming fast, but being able to do just any species, that's a much longer and slower progress, and it requires a lot of habitat restoration and work with living species first.
Is there anything else you'd like to mention?
Oh, well, I have to, of course, say that we at Revive and Restore, we are a nonprofit and we work on donor dollars, donations from anyone in the world. Anyone can go to our website and donate money. Anything from $5 and up helps us. More than 80%, between 80 to 90% of the money that comes into Revive and Restore goes directly to scientists and science teams that do the work. And so if people go to our website, reviverestore.org, they can see a lot of the types of projects we're working on, like saving coral reefs with stem cells and probiotics and genomics for things like jaguars in South America or cloning Przewalski's horses for rewilding someday in Asia. Our reach is global. We have European teams, African teams, Asian teams, South American teams, Australian teams. And I think there's something that just about anybody can get behind and support. And we rely on the support of people like the ones listening to this podcast. So please, absolutely, if you like the work, consider donating and supporting what we do.
Sounds good. I have one final question. What's the meaning of life?
The meaning of life. Oh, wow, the meaning of life. For me personally, the meaning of life is in this life. This one, not the next one, but in this life, to find and spread happiness through the acts that we do with our families, with others, that every life can have a public service, that everything we do can give back to the world as a whole, that we are part of a society and a species that is social, and everything we do can have a kindness and a benefit to other people, to work to remove our angers and prevent hate. That's the meaning of life. And when we think about that, it goes beyond then us as a species to all life on this earth, that we play a role that where we can be a part of an environment. We can be a hunter, we can be a gardener, we can be in the environment and be who we are naturally, and yet still be kind and a part of this world in a good way. Kindness
Ben,
is the meaning of life.
Ben, this was great. Thank you for your time.
Thank you.
Tusen takk for at du hørte på Bendiksen Airways. Dersom du har tips, kommentarer eller vil gi en vurdering av episoden, så gjør gjerne det. Det setter jeg stor pris på.